molecular problem of memory, we go from mind to molecules, and we ask, what are the mechanisms of explicit and implicit memory storage? On the surface of it, you would think that the kinds of molecules used for these two kinds of memory storages will be dramatically different because they are different in three major ways. The nature of the knowledge stored, perceptual and motor skills on the one hand, facts and events on the other. Sites of storage, reflex pathways, cerebellum on the one hand, hippocampus, temporal lobe on the other. And the strategies used, unconscious recall for motor and perceptual skills, conscious recall for explicit memory for specific events in people. This raised the question, do they have any features in common? And can molecular biology, with its ability to reveal homology of relationships, delineate commonalities in what appear to be very, very different memory processes? The fact is, we're fortunate. If we look at these two processes from a distance, we see they do have certain formal features in common. To begin with, both implicit and explicit memory storage have stages. There's a short-term memory, which goes from minutes to hours, and a long-term memory that can last for days, weeks, and the lifetime of an individual. Number one. Number two, we know how to convert short-term to long-term memory. In the simplest case is repetition. Practice makes perfect. And finally, we know that in many cases that have been explored, long-term memory differs fundamentally from short-term memory in requiring the synthesis of new proteins. Are there very different kinds of proteins involved in explicit, implicit memory storage, or are they similar proteins involved? Do they share any features in common? Now, to explore that, we'd like to take experimental systems in which we can study implicit memory storage in enormous detail, other experimental systems, or perhaps the same experimental system, in which we can study explicit memory storage in some detail. The selection of the appropriate model system for a specific problem is one of the great strategic decisions one makes within biology. One is fortunate when it comes to implicit memory storage because implicit memory storage is extremely conserved. You can study this in invertebrate animals. You can study this in flies, in worms, in snails. And you can certainly study this in higher animals like mice and rats and human beings. I'm going to describe studies carried out on snail, Aplysia, but similar studies with very similar results have been carried out on Drosophila and honeybee. And actually, implicit memory storage in mammals uses almost identical mechanisms. Hippocampal-based explicit memory storage requires an animal that has a hippocampus, which means a mammalian animal. And as Tom pointed out to you, an ideal experimental system for that is the mouse because it provides us the power of a genetic approach. So I'm going to begin with implicit memory storage, and I'm going to tell you something about the snail Aplysia. Now, Tom demonstrated for you his lower body strength, so I can't resist showing you something about my upper body strength. I'm going to show you why Aplysia is such a beautiful animal and why many people like to work with it. These are several Aplysia around. I'm going to pick one up, show you what an absolutely gorgeous animal this is as it relaxes. This is the California species of Aplysia. In fact, you might want to touch it. Just rub its hand very gently. You'll see this is a very nice animal. Would you? And you, all of you performed so well, so you deserve this as a reward. This comes from California. It lives on the beaches of California, and this is true for other animals that live in the California coast. It has reduced its life to its bare essentials, feeding and fornicating. And some individuals are capable of carrying out both simultaneously. This animal is so accomplished because of these various things that it does that it has been publicly recognized. In fact, there's a general consensus among the people who work with the animal that it's the animal that has deserved the recognition, not the people who work with it. But the reason one works with it is not because it's accomplished and not simply because it's beautiful, but because of its brains. Your brain and mine has 100 billion neurons, as you heard from Tom. Aplysia has only 20,000 neurons, so 7 orders of magnitude less. That makes the task of analyzing the nervous system much, much simpler, number one. Number two, the cells are collected in clusters called ganglia, and a single ganglion contains about 2,000 nerve cells, which controls not one, but a number of different behaviors. So the number of nerve cells committed to a single behavioral act can be quite small. Less than 100 nerve cells in the limit. In addition to having a very simple nervous system, the animal has a nervous system that is made up of absolutely gigantic nerve cells. These are the largest nerve cells in the animal kingdom. The largest cells are a millimeter in diameter. You could see them with your naked eye. At least I could see it before I became presbyopic. I could see them with my naked eye. The cells are so distinctive because of their large size and because of characteristic pigmentation and position that you can return to the same cell in every animal of the species. You can identify the cell and study the same cell in a naive animal, an animal that has learned something. Now, because of this, you could give the cells names, specific names. You can call them Tom Check or Dennis or Tom Jessel. We're not creative enough to think of that. So we give them very prosaic names. You call them R something or other, R2, R3, R14 if they're on the right side of the ganglion, and L if they're left side of the ganglion. This simple animal with simple nervous system, large cells, generates a whole bunch of different behaviors. But we have selected the simplest behavior that the simple animal has. What we look at is a withdrawal of the gill. This is an external respiratory organ, which it lies in a mantle shelf, which is normally perfused by seawater, and the gill extracts oxygen from the seawater. It's covered by the mantle shelf, a piece of skin that we've pulled back here in this drawing. That mantle shelf ends in a fleshy spout called the siphon. If you now apply a weak tactile stimulus to the siphon, like that, you get a withdrawal of the gill. This very simple reflex can be modified not by one, not by two, but by several different forms of learning. Sensitization, habituation, classical conditioning, and operant conditioning. I want to remind you, these are the behaviors that the behaviorists, the initial students of behavior, have studied. So Pavlov and Thorndike, this is what they were studying. They were studying all forms of implicit memory, and we can see this in the simplest animal. Each of these forms of learning has a short-term and a long-term memory, depending on repetition. I'm going to focus on one form of learning called sensitization, a form of learned fear. So if a horrible noise were to go off in the corner of this room, it would startle the heck out of all of us. As a result of it, most of us, me in particular, would be very nervous, so a modest stimulus that would have no effect at all would startle me. And so with learned fear in aplysia. If you apply a noxious tactile stimulus to the tail, you frighten the animal. It has learned that it's frightened, and now when you apply the same gentle stimulus to the siphon, you get a much brisker withdrawal than you saw before. Let's see this in the real animal. So we're looking at an aplysia stretched out. We're going to focus in on the gill and the siphon. We're going to apply an extremely weak stimulus so you can see the amplification with sensitization. Weak stimulus to the siphon, you see a modest withdrawal of the gill. Now we're going to frighten the animal, startle it, give a noxious stimulus to the tail. That, of course, causes a contraction in its own right. This contraction lasts for seconds, but you can come back minutes later. The same siphon stimulus now produces a much more powerful withdrawal. We can now compare the two, normal and sensitized, and see how much more powerful the withdrawal is in the animal that is startled. The memory for this event is a function of number of training trials. So if you give a single tail shock, plotting here, change in reflex strength as a function of time. The enhancement of the reflex lasts minutes. But if we give the same stimulus 4 or 5 times, we get a memory that lasts a couple of days. So we have a short-term memory and a long-term memory, and we can now look at the difference between them. We know from other experiments, more complicated cases, that protein synthesis inhibitors block long-term memory. So we can see, will that work in this simple case as well? You apply an inhibitor of protein synthesis, inject it into the animal, or apply it to the abdominal ganglion, which controls this behavior. You selectively block the long-term process without in any way interfering with the short-term process. Moreover, this memory that I showed you lasts for a few days. If you train for several days, you can get a memory that lasts for weeks. So you can really, in some detail, compare short-term to long-term memory. So what we next want to understand is, how does this reflect itself in the neural circuitry of behavior? Can we begin to get to the level that Tom was talking about? So